Okay, so this is a lecture that I really enjoy. I enjoy, all of them, and I've got work that is in a lot of areas, but this is definitely one of my favorite ones. So hopefully you like it, but if you don't, it's short, so haha. Like I say, we're going to talk about taste, smell, and touch. These are, I think it's called no-no beans. I should have looked this up before I did this. Allow me to consult my phone. So anyway, this is, I checked it out. It's natto. I think I remembered it as no-no because that's what this picture makes me think. No. But the more I looked it up, I guess the flavor is different depending on different condiments you put in. It's supposed to be really good for you. It's kind of interesting. Anyway, look it up if you're interested. If you're grossed out by it, don't look it up. It's fine. All right. So the different tastes that we've got 5. Sweet, sour, salty, bitter, and umami. Umami is a new one, but probably you've all heard. If you read a lot of food blogs or something like that, umami gets as much play as all the others. But it was only recently recognized as an actual taste among scientists. But it was discovered way earlier than that. And if you haven't heard about it, you can definitely think of things that are sweet like sugar, sour like lemon, salty like salt, bitter like the taste of sun-dried tomatoes, I guess. They have a bitter flavor to me. And then umami is kind of like this savory component. It's not necessarily salty, but it's kind of close-ish to salty. It's this umami flavor. You get it from beef, mushrooms, tomatoes, not sun-dried tomatoes, but the actual just taste of tomato. These are all things that are considered umami. This, which you've probably seen throughout your life, in grade school and college and stuff like that, where there are different distinct areas on the tongue where taste, it's wrong. It's totally wrong. There's not one particular area where taste is for everyone. Now, what can happen is that you can have, in an individual, you might have an area where bitter does come up for them. It might be on the back of the tongue for somebody. It might be over on the side. So that could be true. But for everyone, we don't just taste these things in these places. You can test it yourself. If you take a little bit of sugar water or something like that and put it on the front of the tongue, the sides of the tongue, the back of the tongue, you're probably going to be able to taste it in the same concentration with the same amount of intensity in all of those different areas, unless you accidentally find a cluster of sweet sensitive cells somewhere for yourself. All right. One thing that we do know is that taste is super heavily dependent on the smell of the thing, what the thing looks like, and also what you expect something to taste like. So if you... I don't like ketchup in any way, but one time there was ketchup on something. I don't remember what it was on, but for some reason I thought it was going to be some sort of jelly or a fruit preserve, which I guess technically ketchup is a fruit preserve because tomatoes are fruits. But anyway, I expected it to be a raspberry or strawberry or something like that. I don't know why. I know they don't really look the same, but maybe it was a low light condition, but it's a terrible memory. Anyway, so I took a bite out of it and expecting there to be this sweetness and tartness a fruit preserve has. And then biting into just too much ketchup, which is something I don't like anyway, was pretty jarring. So expectation has a lot to do with something. If you've ever expected something to be macaroni and cheese, but it was actually maybe potatoes au gratin, and maybe somebody didn't cook their potatoes all the way, so they were still a little bit crunchy. Something's wrong, and the taste immediately is kind of off. Once you understand what's going on, it might actually be good. But in the beginning, the taste is all kind of screwed up, and it might even taste bad because you're not expecting it to taste like what it actually is, or even feel like what it actually is. We can smell a lot more things than we can actually taste, and that's one reason why why taste is heavily dependent on smell. And then also vision, we can kind of get an idea of what something looks like, and it helps us get really excited to eat and be prepared for the flavors that are going to, boom, like come at us and hit us. One other thing that's really cool about taste, and there's actually not an umami on here. I'm pretty disappointed in that. I looked for a long time to try to find one of these pyramids that incorporated umami. Doesn't exist. I don't know why. But it should be somewhere here-ish, or maybe at the other end of the pyramid. So instead of a pyramid, it might be like a 3D diamond shape with umami kind of at the opposite end of where sweet is, if you imagine it like that. Or it could be over here as like another node between salt and bitter. We wouldn't know unless we actually did some testing to see how people perceived the similarities between these flavors. But anyway, so that's what happens with these. People taste sour things, salty things, bitter things, and sweet things, and kind of gauge the similarity of these different flavors. And you end up with this kind of pyramid shape where you can have foods that kind of travel along this axis, and they could go from salty to sweet, they could go from sweet to sour, and then there are things that lie somewhere in the middle, like dark chocolate is somewhere between sweet and bitter. Olives are somewhere between bitter and salty with just a slight amount of a sourness to them, which I agree with that placement for olives. I mean, different kind of olives have a different placement for sure. Castelvetrano olives are amazing. I'm definitely saying that wrong, but these are so good. You should go find some if you like olives. If you don't like olives, probably steer clear. Okay. So, taste buds, and different things like that. You've got these little papillae, is the plural. Papilla is a singular. These papillae are like the little hairs that you can see on your tongue. I would show you, but that seems gross. I'd show you on my tongue, but it seems gross. Let me move my head. It's in the wrong place. It's in it where there's not a good place for it. Okay, there we go. So your taste buds are going to kind of like live on these structures. Go and look at your tongue in the mirror after you look at this, because you'll see all of these different kinds of taste buds on your own tongue. So you've got these velate papilla, and you can think about them like a castle with a moat, right? You've got this big, large papilla back here. It's like much bigger than your typical little hair-shaped taste bud that you think of. Again, I'm saying taste bud. The taste buds are like little tiny holes on these little hair-like papilla, but we always call them taste buds. You know, colloquial. Anyway. So you have these large taste buds, and you can definitely see these. They're at the back of your tongue. So if you go look in the mirror, stick your tongue out as far as you can, look at the back, you'll see these like really large taste buds. It's totally normal. People have different amounts of them. Some have more than others. It's totally fine. And you'll see that they have like a moat around them. Well, what happens with this moat is saliva and taste molecules kind of pool around in this moat and hang out there. Why do you think that makes sense? They're towards the back of the tongue. This is where the back of the tongue almost gets to the point where it like slopes down towards, the esophagus and the larynx. But if we're talking about eating, it's going to be going towards the esophagus. Well, it makes sense that you want to have this moat around it to hold the flavor in as you get back to this area, because here's where things can start to like trickle down You should have your tongue cupped, which is in some of the other lectures in the section. While you're chewing, your tongue is cupped. But then these velate papilla are back here, so they're not really getting a lot of the flavor. But then as you go to swallow, the tongue curves and it's going to flow over them quickly. The moat is there to trap some of the saliva and flavor molecules so that you can actually taste it in that part of the tongue. There are also taste buds all the way down the tongue. They're not in the same concentration that they are on the top of the tongue, but all this crazy structure back here does have taste buds in it. And even the very top of the epiglottis, the lingual surface of the epiglottis, has some taste buds on it too, which is kind of weird and interesting and cool. But they're sparse, right? So you're not really tasting a ton. Anyway. All right. Actually, okay, quickly, I guess I should say, one of the things that those taste buds on the lingual surface of the epiglottis do is that they will help your body know when it's time to swallow. So if there is a sensation, if it picks up on some sort of flavor that might be trickling down accidentally, it will signal a swallowing reflex. So there's a whole bunch of things that should be triggering a swallowing reflex. So just the fact that there's a sensation of liquid moving back there, that should help trigger a swallowing reflex. Taste sensation triggers a swallowing reflex. The fact that the food is backwards in the mouth on the back of the tongue, touching the facial pillars. There's a ghost in here. There we go. This should all trigger a swallowing response, but it all kind of works together to trigger this response. All right, so that's velate papilla. Then you've got these foliate papilla, which actually means leaf. It's a leaf shape. And they're kind of on the sides. They're a little bit larger, very vaguely shaped like a leaf. Again, mostly on the sides. Your fungiform, these are the large ones that are kind of placed randomly on the surface of your tongue. So if you just stick your tongue out, you'll see like the little ones, and then you'll see these like large ones interspersed totally randomly. Those are the fungiform. That means mushroom shaped or mushroom like. And so they kind of, I guess, look a little bit like a mushroom. In fact, if you look at the fungiform papilla, that is what's inside of the velate papilla. So the velate has the moat around it. It's essentially a fungiform papilla with a moat around it. Or the fungiform is the velate without the velate. It means valley. So without that, it's just the fungiform. And then your typical taste buds are going to be these filiform papilla, which just means hair. They're really small, kind of wispy. And that's your typical taste bud. So how does taste actually get in? Well, right here, you can kind of see this. I should have a blow-up. Well, I do have a blow-up. What am I talking about? So you've got these little openings in these papilla, these tiny little openings here. And here's the blow-up. So this is the surface of the papilla, and then you've got this hole. This is called a taste pore. And so molecules, taste molecules and saliva are going to go into the taste pore, and then inside the taste pore you can't actually see it because of the background here is black, but there are these tiny little hairs. You can see the bottom of them right here. They should be sticking up in the taste pore. These are the microvilli, and they are just like sensing chemicals. They're going to pull, I say unidimensional chemical makeup because a microvilli does not sense multiple tastes. A microvilli senses sweet or bitter or something like that. Or even like very fine-grained distinctions within those large ideas. So you'll have one that just looks for like certain chemical structures that makes things taste sweet. So anyway, if it detects that chemical, it becomes a neural impulse in this cell right here, and then goes on, all the way up to the brain. All right. So where is taste processed? Well, that's a good question, isn't it? So a lot of different sources point to the thalamus. Which the thalamus is the main relay of sensory information in the body. So it does make sense that it's going to go through the thalamus. Does it stop there? Maybe, maybe not. Other places point to the amygdala, but the amygdala governs strong emotions. And so it makes sense that some flavors are going to govern strong emotions. I know that when I'm hungry, well, even when I'm not hungry, but I have something that tastes good, that's a pretty strong emotion. And so you might end up with secondary activation of the amygdala, but it's not necessarily because of the taste. It doesn't mean taste is processed there. It means my enjoyment of the flavor is processed there. It could be frontal regions, because that's where we're thinking about the flavor. But again, that could be associations with the flavor and not the flavor itself. The insula, which remember is the fifth lobe, that could be on the last optional final as well. So the insula is the fifth lobe. It's the center of visceral emotions and visceral physiology. Visceral, again, is like the things that you actually feel, especially coming from down towards the stomach. So things like disgust that makes you want to vomit. Sometimes types of fear will be a visceral emotion. But also hunger and taste might be visceral emotions as well. And so it's possible that the insula is responding to taste as well, which is what this picture is showing. I don't put a lot of stock into this picture because, I mean, who knows when this is from? I just thought it looked cool. But anyway, it could be processed in the insula. It could be that the insula might be a primary site and then it's passed on to reward centers beyond there. We know, for instance, that chocolate activates the reward pathways that are common to things like listening to music, getting money, sex, all sorts of different things like that. They all kind of operate on the same reward pathway, right? So it could be that taste is processed in the insula and then it goes to the reward pathway. And like I said, it could be that the signal is separated at the thalamus and then it goes to the insula beyond there. But it probably, almost certainly, goes through the thalamus. Whether it stops there or uses that as a relay is kind of not a question that's been answered yet. So how many actual taste dimensions are there? We know there's 5 individual tastes. And if you're somebody who reads a lot of, like, if you drink a lot of wine, you know that, like, wine aficionados think that there's, like, 80 tastes. And they'll say things like, fruit notes, this was probably grown in the shade of a mulberry bush. That's what it tastes like. And you're like, how do you know it's grown in the shade of a... Anyway. I've done some more perceptual work with this. I used beer. I enjoy beer more than wine. And we ended up showing that there's 2 primary dimensions to beer. Each one has a different main taste. So each end, I should say, has a different main taste. So there's 2 primary dimensions, but there's one end. There's 2 ends to each dimension. What the hell am I talking about? What am I talking about? So let me consult the one that's written on my whiteboard here. So we ended up seeing that there was a bitter and a sweet and a light and a heavy. So bitter, sweet, light, heavy is what we ended up with for our beer research. That's kind of a pilot study. We're still kind of looking into it. Here's a couple different things that we've seen. So here's a different group that's done work with beer. They break it into a ton of different flavors. I'm not sure what drinkability tastes like. Does that really seem like a taste dimension? Not to me. Here's one that breaks down coffee into a couple different flavors. All right. So aging can lead to taste desensitization. This is just something that we know. It's kind of an unfortunate thing. And this, I mean, if you have an older parent or a grandparent, you should ask them about this. My mom always complains about burgers. And she's like, burgers never taste like they do in the 70s. She thinks it's something to do with, like, the meat industry. But I'll take her to these places that have... Oh, my face is in the wrong place again. I'll take her to these places that have, like, grass-fed burgers and all this crazy beef. She grew up on a cattle ranch, like a beef producing ranch. And so like we've gone to places that produce their own beef, and I'm like, how's this? And she says, well, it just still doesn't quite taste like it did in the 70s. And I have to wonder if, I mean, she thinks they're closer, and like certainly probably McDonald's meat is trash, but and where these other places that I've been taking her to are better. But I also think that there might be some of this age-related taste desensitization going on. Why does the thing not taste as good as it did in the 70s? Well, the taste buds are just not as sensitive as they used to be. There are also disorders that can lead to diminished taste in, like, aside from aging. So you can have a younger person with some sort of certain types of TBI, and I don't mean like different types of TBI lead to this. I mean like different areas where any sort of TBI occurs can lead to desensitization of taste. You can also have nerve disorders that lead to desensitization of taste, or aging, or a combination of the two. Like somebody might be older and then they have a stroke and then taste is impacted. But what's really strange is taste isn't impacted the same. Like all of these different tastes aren't impacted the same. So what's going on this graph is that this one is stronger. This is with age. So the dark bar is showing that the taste got stronger. The gray bar is showing that they're the same. And this light bar is showing that the taste got weaker. And what is really terrible, because I like sweet things, is that with age, it seems that sweet things get weaker for over a quarter of the population. There's another quarter of the population that, no, sorry, this is number of patients. Oh no, that's even worse. This means over half of the people in the study reported that sweet was diminished. Less than half of them thought it was unchanged. Only one person thought it got stronger. Why? It would be great to be that person. And then you can see that salty is the next one to go. Again, I love salty things. Sour, then bitter, and then metallic. You know, so I guess if you don't like the taste of pennies, good news. Some of you will have that taste diminished, though not nearly as many as with sweet. Most people didn't have their metallic taste change at all. This is kind of a weird one, though. Bitter tastes seem to get stronger with age, more than, compared to all these others. So it's interesting how the tastes kind of it's not like taste diminishes altogether. It's kind of like the balance shifts, which is really strange. It could be that people eat more sweet and salty things, and then because of that, those are kind of overtaxed, and those pathways are overtaxed, and so they get kind of habituated over time. And then things that you don't eat as much, like pennies and bitter stuff, seem to get stronger, or stay the same. Could be. I'm actually not sure. It's a really interesting area of research. There's not a lot done on aging and taste. So if you're interested in research, interested in this research, this is a really cool area to go into. Smell I'm not going to talk about a ton. I just want to touch on it because it also affects taste. The odor molecules are one of the things that interface almost directly with the brain. So you can see that here's your cribriform plate right here, and the olfactory bulb lays right on top of it, and then you've got these olfactory receptors that come right down into the nasal cavity. So your smell is interfacing pretty much directly with this. This would be kind of like a ganglion right here. So then this is a huge pathway that's going to go right to the thalamus. It's pretty cool, pretty interesting to me how almost direct that pathway is. Again, down here, protruding through the cilia of the nasal cavity, protruding through the skin, the epithelial layer of the nasal cavity, you've got cilia. This is a lot like the microvilli that are in the taste pore. So taste and smell are actually very similar if you think about it. They're both chemoreceptors. They're both picking up on different chemicals. They both use these little cilia in the nasal passages, microvilli in the taste buds, but they're still like little hair-like structures that respond to chemicals and then produce a sensation. And they're also tied together perceptually, right? Because we know that smell affects what things taste like eventually. So, pretty interesting to me. That's all you need to know about this slide. Okay, and then there are some people that can detect a lot of smells. If you are a fan of Psych, you know that Burton Guster is a super smeller. And then I'm also related to a super smeller. This is my mom. She can detect all sorts of things. So like if I'm in a driver somewhere and I have like a new like car freshener hanging from my rearview mirror, she will not ride in my car because she's too sensitive to the smell. So it's I guess it's kind of a cool superpower to have, but it's not always cool for the people around you that have to deal with it. So anyway, a human can detect up to about 1,000 different smells, and dogs have almost 9 times that. So 9,000 smells, 9,000 different smells. Imagine the crazy scent world that they live in. Here's one clinical concern that I didn't add into the clinical area, but sometimes post-laryngectomy smell is diminished. The sense of smell is diminished. I want to point out, though, this is not because the sense organ, none of the olfactory bulb, the cilia, none of it is actually affected during the laryngectomy. And yet after laryngectomy, people will complain that they can't smell. Well, the reason is, after laryngectomy, this is where the air comes in. You're breathing through this tube, not through your mouth. And so if you're breathing through this tube, the air is coming in. The air is not passing over the nasal passage. So, of course, the sense of smell is diminished. No air is coming into the nasal passage to carry the molecules in. So what you can actually do if you have a patient that is suffering from this is you kind of reteach them to smell by yawning. So it's called the polite yawn technique. So I guess if you're in a meeting, you don't want to be like, so tired, shut up. But if you do have to yawn, you'll yawn through your nose, right? So you kind of do the, and try to cover it up as just taking a sniff of your coffee, I guess. I don't know. So you reteach people how to do this yawn. So they kind of yawn on command, and then they'll yawn through their nose on command. And what this does is, and it makes sense, it draws air into the nasal passage, and then it allows them to be able to smell again. So they can have a laryngectomy and breathe through here, but then teaching them can also move the air up to here and allow them to fully sense their world like they want to. Also, related to this, there's some research that points out that loss of taste that I was talking about before might actually be related to a loss of smell. So it could be that the cilia in the olfactory bulb are a little bit more sensitive to degradation over time than the papilla and the taste buds. And so it could be possibly, it's a new area. I think this study came out 2 years ago. I mean, in 2018, not necessarily 2 years ago. I think it was late 2018. So there's a lot of work left to be done in the area. But it could be that it's all smell related and not taste related. But again, it's really hard to separate smell from taste because they're so interdependent on one another. Okay. There's a lot of touch sensors in the mouth. So also, this is something I want to point out. We haven't talked about this for a long time. This is like way back in the neuro slides, right? This is the homunculus again. The last time you saw him, he was kind of like laying over the top of the brain, representing the different areas, so the different parts of the motor cortex correspond to roughly the shape of the human body. And this guy was showing you how much proportionally of the body is represented by different portions of the motor cortex. So if you look at the mouth on this guy, you see that there's a lot of representation for the lips, the tongue, all sorts of stuff like that. So there's a huge representation of touch, mouth-based touch in the somatosensory cortex. One way that you can do this at home, you can prove this to yourself. Chopsticks. You don't have to use chopsticks. You can use pencils or pens, or if you're like a total daredevil, you can use knitting needles. Is it called the 2-point discrimination test? So what you can do is close your eyes, and you'll have someone else do this to you. So you're going to take these 2 points. The first one you want to do is on the back of your hand. And what you want to do is put these 2 points very close to one another. You've probably done this as a kid. I did it as a kid. You can put the 2 points next to each other and then start moving them apart little by little. Remember, your eyes are closed. Someone else is doing this to you. And then you want to tell the person when you can sense there are 2 things touching you instead of 1. So at first it'll feel like there's just 1. And the further you move it apart, you'll feel like there's 2. Then I want you to do it on either your upper arm or your back. They're going to be very far apart before you can actually tell that there are 2 things. We don't have a lot of representation in the somatosensory cortex for our back. We do for our hands. You can see the back is pretty small relative to the hands. The hands are large. The back of the hands are actually smaller than the front of the hands. So then what I want you to do is flip it over, do the palm, and then do the tongue. And it's actually, these might not even be sharp enough. I can sense the part where they come together. So these are too big. Like I can sense that they're 2 things the whole time. So these are actually too big. On the hand, yeah. So this is working the way it should. The back is going to be the furthest apart. The back of the hand is going to be a little closer. The front of the hand is going to be quite a bit closer, and the tongue is going to be very close. If you're using chopsticks like I am, it's not even enough. Like I might have to use, yeah, just barely before they actually touch each other using these like thumbtacks. Then I can get down to not feeling that there's like 2 individual things. So go ahead and try that. It's kind of fun. That's just showing how absolutely receptive, the cells in the mouth are. You've got a couple different things going on for touch receptors in the mouth. You've got these Meissner corpuscles, and they're looking for pressure. And actually, the Meissner corpuscles, you can see them. This is kind of a depiction of them here. They're looking for pressure. On the roof of the mouth, you've got Meissner corpuscles as well. And so when something pushes on the roof of the mouth, like if you put a cracker in your mouth, that'll begin the chewing reflex. Now, yeah, you can chew on your own. You don't necessarily need a reflex to tell you when to chew. But this will kind of put all of the muscles into alignment to get ready to chew, just pressure on the roof of the mouth. So it kind of gets the reflex ready to go. You've got Merkel cell receptors as well. These are these little guys here. They're responsive to light touch. They're also receptive to about 15 hertz vibration. They're right here on the lips. There's a large amount of them that are here on the lips. And actually, this is what is responsible for sensing the touch of infants' lips. And that starts the rooting and sucking reflex. So the touch of on the Merkel discs down here. You've also got Pacinian corpuscles that are receptive to vibration, and Ruffini endings that respond to stretch. The 2 that are most important are the Meissner corpuscles and the Merkel cells. So the thermal cells kind of respond to the change from the overall ambient temperature in the mouth. And this would explain why your coffee, the temperature of your coffee that you sense is dependent on not just the temperature of the coffee, but the temperature of the outside, the temperature of your mouth. So if you just go on a run in the winter, it's near freezing, and so you're breathing through your mouth because you're trying to pull in as much air as possible. Your mouth gets pretty cold after that. You can drink something that's not super hot, and it seems pretty jarring because these temperature receptors are just kind of looking at change from the overall ambient temperature. It also, let's see, what do I want to talk about here? Well, your cold receptors have more range than heat, which kind of makes sense. The cold has different gradations that it'll sense, and heat just kind of goes from like a little warm. This is too much. We're in pain. So there's more of a range to cold than there is to hot. And then also, I'm going to skip over this and want to talk about this. So, well, no, that's what I just said. The hot turns into pain pretty quickly. So you've got nociceptors, which are actually pain receptors here. Oh, yeah, I've got a good picture of it here. So you've got these pain receptors here and heat receptors here. And so there's a lot of work going into the cold receptor. There's a bunch of different cold receptors for different amounts of change for cold things that you're drinking. There's a lot of space before the cold turns into nociception. And I mean, for some people, that range is larger than others. This gets into, brain freeze territory is when the cold receptors have to turn their work over to the pain receptors. The heat receptors will turn their work over to the pain receptors much more quickly. So it's like warm, hot, pain, the end. What's really interesting here is the capsaicin chemicals, which is in peppers, different peppers. If you like hot foods, all these peppers have capsaicin in it. So those are actually not processed any differently than regular pain. So the nociceptors just kind of pick up on those chemicals and say like, oh, we're being burned. And that's where the sensation of like spicy food comes from. It also may indicate why hot things burn your mouth in the same way that really hot temperature things burn your mouth in the same way that hot spicy things burn your mouth. The sensation is kind of the same. It's kind of this overall just like flare up of pain. And it might also be why we kind of conflate the idea of spicy with hot. Some people say like, is it hot? It's like spicy hot or heat hot. They have to make the distinction because perceptually they're very similar. One of the reasons why we might like spicy foods, why some people might like spicy foods, is that when you have pain receptors firing, then the body releases epinephrine. There's a circuit in the medulla that when there is pain, especially long-term pain like a chemical burn in your mouth caused by one of these great chili peppers here, then epinephrine kind of floods the body. What's cool about getting a chemical burn from a pepper is that that you're not actually being damaged. You know what I mean? So like if you got a chemical burn on your hand from a chemical in chemistry class, this is a bad thing. And so you will have epinephrine released to try to, calm you down and counteract all the bad things that are going on. But there is a problem, and it's a problem that you have to deal with. If you break a bone, epinephrine is released, but again, this is a problem. This is a bad thing. Spicy food is kind of ingested, and you think it's like, oh, it's a fun thing. I know it's just food. It's not going to kill me. And then you get the epinephrine that's released, and then you kind of get this relaxation that comes after you eat the spicy food. So there's a lot of research that points to people who are stressed tend to like spicy food more, or spicier food, and that might be because they get more of an epinephrine hit, then they can relax a little bit more. Also, if you watch Hot Ones, some people, when they eat some of the hottest wings, they'll say like, gosh, I feel drunk now. And that could be because of this release of the chemicals to try to counteract the chemical burn in their mouth. So anyway, this could be why me and some of you like spicy foods. All right, that's the end of this lecture.